check audio, because I suck at audio. But it's really like a fan is going off on me. Okay, is that better? Oh. Do. Oh, how's that? No. I wonder if it's just the mic. What if I switch to that? Does that work better? No. Well, now you can't even hear me. I don't know. It's still like my fan's going off. How's this? Does that sound better? That can be heard. If I change it. No, not what I want. Not what I want. Is this better? Is this better? No. None of it's better. Okay. I think that is better actually. I think that is better actually. Alright. Sorry. I'm sorry, guys. Anybody watching? Nobody's watching this. I'm sorry. Anybody who does, I am sorry for that silliness. Anybody who does, I am sorry for that silliness. Anybody who does, I am sorry for that. But now it's looping. Why is it looping? Is that better? Hello? Okay, no, it's not looping. Oh, feedback. Okay, hello. Ah, this is the number one reason that discouraged me from streaming before. So, welcome everybody to my channel, Bones of Amber. And let's go over some new articles. All right. So, oh, I guess I need to minimize this so that you guys can see. Uh-oh. There. And you can see me. And you can see the article, which is the important part, not me. Well, yes, there we go. All right, let's get started. First article is from John Hawk's online blog thing, Denisovan, HLA, and its role in immunity. Today's people have inherited several immune alleles from archaic people. Understanding what difference they made is a continuing challenge. Everything I write about function in archaic genomes starts with immune phenotypes. As early as 2006, my research was focused on understanding how adaptive gene variants from ancient populations like the Neanderthals may have been important to the evolution of later people. Even in that early work, I saw that immune system genes were among the best candidates for today's people to have inherited from archaic groups. Oh, what's this? Subscribe? No, thank you. I do follow you on Twitter, though. And you should. He's very active on Twitter. It's quite nice. I was drawing my comparison from non-human species like cattle, in which geneticists have documented gene flow across distantly related populations, a phenomenon called introgression. Some of the most common introgressed genes were involved in the immune system of those animals. It's easy to see why. Pathogens often find ways to hop across natural boundaries, even the boundaries between species. When they do, the pathogen's new hosts may benefit from gene variants that evolved in other populations that co-evolved with similar pathogens. When Svante Paabo's research team first recovered ancient DNA of Neanderthals and Denisovans, established researchers in immunogenetics quickly started looking at these genomes. One of the first studies to investigate possible functional effects of Denisovan and Neanderthal genes was published in 2011 by Laurent Abbey Recht and collaborators, who looked at the large family of genes known as human leukocytes antigen, HLA. In other vertebrates, these genes aren't called human, instead they're known as the major histocompatibility complex, MHC. I'm coming to the stage of writing in my current book project that I have a lot of background research to share. I thought I would start by sharing some things about Denisovan DNA and its role in immunity. The role of HLA. The immune system is complicated. A human genome has more than 20,000 protein-coded genes, and research has connected more than 1,500 genes to immune responses. That's at least 7% of the genome. This complexity evolved not just during the 7 million year evolution of hominins, but across the hundreds of millions of years that vertebrates have diversified. Today, immune systems help humans survive more than 1,000 viruses, bacteria, fungi, and parasites that infect people around the world. Most of the body's cells use HLA class 1 molecules to help transport and display small fragments of proteins known as peptides in their cell membranes. One class of immune T cells, known as cytotoxic CD8+ T cells, interact with the HLA class 1 molecules. When they detect a foreign antigen, like the kinds that are produced by viruses, the CD8+ T cells kill the infected cell. Illustration from OpenStax showing the pathways of MHC class 1 and 2 molecules within an antigen-presenting cell and their interaction with the T cell. T cell receptor, the T cell MHC proteins now carrying antigens are released from the vesicle and travel to the outer surface of the cell membrane. Cool. So it detects it, releases it. The dendritic cell is now presenting antigens, which will activate T cells that bind with the MHC protein. Look at that. Another class of immune cells known as antigen-presenting cells, APCs, specialize in taking peptides from outside of cells. They combine these peptides with HLA class II molecules and then present them to CD4 plus T cells. The helper T cell, the help is that they activate another kind of cell known as B cells to produce antibodies. Antibodies are large Y-shaped proteins that can bind directly to viruses and bacteria, inhibiting them from infecting cells. Antibodies also tag the pathogens to help large immune cells called macrophages to engulf and destroy them. Diversity is central to the function of the HLA system. Each of the genes has many alleles in human populations, and individuals with rare alleles sometimes have a big advantage when new pathogens strike, a pattern known as frequency-dependent selection. Having two different alleles for the HLA gene can sometimes provide dual protection against different pathogens, called heterozygote advantage. Both these patterns are known as balancing selection, and they can favor new alleles and haplotype combinations that gene flow brings into a population. That includes genes from archaic human populations. Studying haplotypes. For a particular gene, such as HLA-A, the different forms that can be detected are usually called alleles. The different genetics states of any polymorphism in DNA, including single nucleotide polymorphisms, likewise are usually called alleles. Nearby alleles on the single copy of chromosome are generally inherited together, unless recombination during meiosis has happened between them. The co-inheritance of nearby alleles means that geneticists can identify long chromosome segments transmitted over time through family trees, known as haplotypes. Haplotypes can span one, several, or even dozens of genes. They get shorter every time as recombination whittles them away. DNA from archaic people is divided into a patchwork of haplotypes and vary in length from 20,000 base pairs up to 1,000,000 base pairs or more. Most of these chunks are rare today, but a few have become quite common. The fraction of Denisovan ancestry across the whole genome maxes out at around 4% in some indigenous groups of the Philippines, and up to 3% in the indigenous peoples of Papua and Australia. In mainland East Asia, the fraction is much smaller, only around 0.2%. For other Eurasian populations, the amount is even less, often 0.1% or smaller. Even that tiny fraction still adds up to 6 million base pairs across a genome. But as the fraction gets smaller and smaller, it's very hard to be sure that DNA comes from one archaic group, not another. Each of the potential ancestral groups was itself diverse. That means incomplete lineage sorting within the ancient African population or ancestry from Neanderthals might resemble Denisovan ancestry in the case of HLA variation. Balancing selection was also elevating diversity in each of the ancestral groups. Denisovan HLA variation. In their early work, Avi Racht and co-workers highlighted HLA haplotypes in today's humans that may have come from Neanderthals, Denisovans, or potentially unknown groups. The ability to assess HLA haplotypes in archaic genomes was limited by the highly fragmented state of ancient DNA. Still, they could look for the allelic differences within a nearby specific HLA gene and suggest which human HLA haplotypes matched the archaic DNA. In the Denisova 3 genome, they suggested that the HLA-A2 allele and the HLA C 1202 allele were present. These two alleles appear in linked form as a combined haplotype today in Papua New Guinea and adjacent areas. The other alleles that seem to be present in the Denisova 3 genome are HLA-A11, common today in East and Southeast Asia, and HLA-C15, common in Papua New Guinea and Australia. We've got a nice map, geographic distribution of HLA-A11 and HLA-C15 alleles in present-day human populations, both inferred to have been carried by the Denisova 3 individual. Image accredited to the Abiratchidin co-workers. Let's see. Oh, nice. Okay, so the -C15, nice hot spot here. And then, of course, down in Australia. And then A11, you have a pretty good hot spot for down in Asia. Well, looks like a little bit of people up in here too. Not many, but still there, separated. That's funny. I wonder why these are blocked out. Just no data for it? The linkage of alleles upon longer combination haplotypes is an important part of the biology of the HLA complex in humans. It can make studying these genes fiendishly difficult. I remember tracing across, not me, Fox. I remember tracing across the first Denisovan genome on my computer screen, looking at sequence polymorphisms in today's genomes to see if they were covered by the short fragments of Denisova 3 sequence. My then PhD student, Aaron Sams, whose dissertation work focused on the evolutionary history of autoimmune diseases in humans, spent a lot of time with HLA and archaic genomes. It proved to be a big challenge. Specific identification of alleles is very hard to achieve for ancient genomes with their large number of gaps and missing data. These limitations have shaped the history of research on archaic HLA. For example, Abiratch and coworkers proposed that the HLA-B73 haplogroup was a strong candidate for Denisovan ancestry. Their idea did not come from the key mutations that define HLA-B73 itself, but from linked mutations that are often part of the same haplotypes as HLA-B73 in living people. Later work has suggested that HLA-B73 was likely passed down from the African ancestors of today's people, not Denisovans, although it is still possible that some linked haplotypes had a Denisovan origin. Okay, so you can't get the full genome in ancient DNA. You get patchy results. You don't always get all of the base pairs and stuff, but from what they're saying, they're able to get certain mutations that are linked today with this specific haplogroup. And since those mutations are within this haplogroup of today, they think it's linked to the Denisovans, but then could have been passed down from African ancestors of today's people. So, because Denisovans are up in Europe and like Asia and everything. Wouldn't that mean that the HLA-B73, if it is also linked in African ancestors, where's the connection to the Denisovan side? But with HLA genes from Denisovans, the lack of ancient sequences remains a fundamental limit on our knowledge of variation in the past. There may be many HLA haplotypes across East and Southeast Asia, or even into the Americas. I lost There may be many HLA haplotypes across East and Southeast Asia, or even into the Americas, Europe, and the Pacific, that we might recognize as Denisovan if the DNA of southern representatives of that population were known. Frustrated about function. The search for the archaic DNA's role in our immune system is a frustrating puzzle. We can find a lot of associations for specific alleles in modern people affected by disease, but we rarely know how these ancient DNA fragments helped our ancestors fight off ancient pathogens. While we have many ideas about what Denisovan HLA genes do, very few studies can pinpoint which pathogens they actually fought or how they worked. Part of the problem is the sheer number of possible HLA alleles. HLA molecules hold out peptides for the rest of the immune system to see. With dozens of different HLA alleles and thousands of different pathogen peptides, it's very hard to figure out which combination is most important for a disease without studying a huge number of people. A bigger problem is a shortfall of investment in investigation of both human populations and their pathogens. Today, the people with the most Denisovan DNA live in island Southeast Asia and Oceania, but historically, these regions have been overlooked in genetic studies, with fewer than 1% of participants in genome-wide studies coming from these areas. I wonder why. Meanwhile, the diseases that may have most affected ancient people are also understudied. Some are so underrepresented in medical research that they are officially known as neglected tropical diseases. Only a trickle of research dollars goes into conditions like yaws or scrub typhus, or into parasitic conditions like filariasis and roundworm. These and other diseases that are persistent in soil, foods, or fecal matter would have been sustained over time by repeated reinfection in small-scale societies. But their interactions with the immune system are less well known than many diseases that are more important in industrialized nations of Europe, East Asia, and North America. What this all means is that the discussion section of papers about Denisovan HLA and immunity more broadly usually have statements that look a lot like arm waving. A quote from Abby Recht. HLA-A11, an abundant archaic allotype in modern Asian population, provides T-cell mediation protection against some strains of Epstein-Barr virus, and in combination with a peptide derived from EBV, is one of only 2 HLA ligands for the HIR3DL2 NK cell receptor. I just love how they name these things. There's always like a meaning behind these things, but if you don't know, it just looks like gibberish to people. It's valuable to note such indirect evidence about possible functional effects of Denisovan alleles. The Epstein-Barr virus, in particular, may have had impacts on past human groups, but what's frustrating is how little else can be done with today's data sets. Even in this case, exactly how the course of EBV may have been affected by the possible Denisovan allele is unclear. Some research groups are taking a broader look at the function of haplotypes from archaic people by studying their effects on gene regulation. This is exactly the kind of investigation needed. In a recent study, researchers led by Danit Yarmakovich found a strong link between Denisovan HLA haplotypes and adaptation to lowland environments in Papua New Guinea. They studied the genomes of over 50 people from the highlands and more than 70 people from the coastal island. The island group, which has likely faced different pathogens, had a much higher frequency of Denisovan haplotypes, particularly in the HLA region. It's a reasonable explanation for this pattern that diseases of the coastal lowlands may have selected for greater immune diversity. The research into archaic DNA's role in our immune system is still in its early stages. To fully understand how ancient genomes matter today, we need to broaden our focus. This means studying more of the world's diverse populations that are underrepresented. Wow, I cannot say that word today. Underrepresented
in recent research. It also means investing in research on the infectious diseases that have shaped human evolution for millennia, even if they aren't the hot topic for research in the global north. The answers to some of the biggest questions about human health may not be found in our cities, but in the echoes of ancient migrations and the enduring battles against disease of the distant past. As scientists continue to explore these genetic whispers from the past, we're not just uncovering the secrets of an extinct human relative, we're finding clues about our own health, revealing the deep and complex connection between our long-gone ancestors and the changes our bodies face today. This is interesting, and I am looking forward to your book, whenever that comes out, that you mentioned. So that'll be cool. Interesting. Next, moving on. Thank you, John Hawks, for that wonderful article. Let's see. I gotta pay. At least we can do the abstract. We can see the abstract, and if anybody wants to pay for it, subscribe. 119 a year. Or if you are still in school or you are working at an institution, you can actually access it through your credentials on that. I am not currently in school or teaching at a school, so I don't have that institution access. Artificial intelligence, AI. Emerging uses of artificial intelligence in deep time biodiversity research. Isn't AI like the hot topic right now? Everybody's talking about it and worried about what the future holds. So it's very interesting to see how it improves. I'm sure everybody's been seeing the like artwork and everything on it, and that's really cool. But I think while being able to produce simple, easy works of art, especially if you aren't an artist, AI in these type of contexts can do a lot of work and help researchers and save a lot of time, honestly, because combing through your DNA can take a lot of time, and having the assistance of something like artificial intelligence can shorten, although I'm sure everybody is aware there are a lot of issues with AI as well, and how accurate it can be. So AI is transforming scientific research by enabling the analysis of vast and complex data to address long-standing questions. Here, we offer our perspective on the applications of AI in paleontology, focusing on how AI can change our understanding of biodiversity through deep time. We began by exploring current methodologies used to automate fossil data processing, analyze morphologic traits, and model evolutionary patterns. Then we discussed the limitations and challenges of data availability and quality, that the extent to which AI-based models can capture biological and geological processes. Additionally, we highlight future avenues for exploiting AI modeling that have the potential to transform future research. Finally, we emphasize the importance of ensuring that the benefits of AI do not exacerbate existing inequalities in computing infrastructure. We emphasize the importance of ensuring that the benefits of AI do not exacerbate existing inequalities in computing infrastructure. What does that mean? If AI can improve deep time biodiversity research, such improvement must be made available to the entire research community. Although AI has the potential to propel paleontological research into a new era of discovery, it remains a relatively new tool, and its full capabilities have yet to be realized. Engaging with the challenges and opportunities presented by AI is essential to ensure inclusivity and progress in the paleontological research community. But engaging with the challenges and opportunities presented by AI is essential to ensure inclusivity and progress. What does that mean? I don't know. I wish I had the full article, because now I didn't know what they mean with it. We got the references. Are there any figures I can see? Ooh. Existing potential uses of artificial intelligence to advance understanding of deep time biodiversity. Can I? Ah, you suck. Can't look at anything. It's got a whole bunch of stuff, but that sucks. I only wish I could read it, but I'm not paying 119 to be able to access the article. Sorry about that. If you're interested, it's a Nature Review Biodiversity title Emerging Uses of Artificial Intelligence in Deep Time Biodiversity Research by Danielle Silvestro and Catalina Pimiento. Yes, if you have access or are willing to pay for it, that is it. Moving on, Smithsonian Magazine. Through much of human history, women have shaped evolution through food processing. An anthropologist highlights the revolutionary role of the practice, which was often led by women and was just as crucial to surviving and thriving as hunting. What is that? It doesn't say what that is. All right. It's the rainy season on the llanos of south central Venezuela. Except for small rises of high ground, the savannas are flooded under several inches to several feet of water. Game has migrated out of the area, and fish are dispersed and difficult to find, but underground tubers are plump. Oh, okay. My husband and I have been living with the Pume South American hunter-gatherers for some months. On a hungry morning, we accompany a group of women and children, walking single file to where they know the tubers are easy to dig out of the soggy, shallow sand. Even young girls under the age of 5 have their own small baskets to fill with the thumb-sized tubers that are the main source of food this time of year. Back at camp, the men who had gone hunting and fishing all day had no luck. The women and children divvy up tasks, peeling, slicing, and soaking the roots to neutralize their bitterness. Later, after they are roasted soft in coals, people share the food out across hearts, hunger sated for another day. It is a beautiful picture. I like that picture. Pume man and woman hunt and gather food on the Venezuelan savannah. That's just a really cool picture. I'm an anthropologist who has studied how women and children spend their time in hunter-gatherer and subsistence farming societies for the past 30 years. Over the decades, it has puzzled me why meat has dominated the story of human evolution, with men as the prime mover shaping our biology and behavior. My research, alongside the work of many colleagues, reveals the central role that women and children play in getting food, usually processed in some way on the collective table day in and day out. Honestly, it was probably central because it's such a punch of calories with meat, and then kind of fascination, I guess, or preoccupation of being able to dominate a landscape and other animals. Dominate may not be the best term there, but it gets my point across. And usually, like you said in this part, it's relied upon during times when food is scarce. I'm curious if they had the choice every day of meat or the tubers, if they would choose the tubers very often. I don't know. A brief history of food processing. While human diets vary widely across cultures, food processing is common to them all. Other than some fruits, berries, and leafy greens, most foods in traditional diets worldwide are processed in one way or another. Cracked, ground, pounded, grated, sifted, winnowed, mashed, peeled, smashed, bleached, shelled, plucked, descaled, deboned, butchered, boiled, roasted, or otherwise cooked. Taking these extra steps gave humans a survival edge when seasons were lean. Favored foods were depleted, or when home ranges became constricted because of conflict or population pressure. Processing food broadens and diversifies our diets, allowing us to thrive in a wide range of environments. Over the last 3 million years of evolution, breaking food down into small absorbable bites shifted from a primarily gastrointestinal process to to an external process. Going to this trouble had a lot of benefits. Many foods humans consume are indigestible or toxic without being processed. Cassava, also called manioc or yuca, a staple in South America, Southeast Asia, the Caribbean, and Africa, contains a cyanide derivative that is noxious if eaten raw. Other common vegetables, such as eggplant, potato, beans, kidney, castor, lima, and cocoa, nuts, such as bitter almonds, palm nuts, and seeds, such as cicade seeds, rice, are either lethal, toxic, or taste bitter. But once powdered, soaked, ground, bleached, fermented, or cooked, they become palatable. Wheat, barley, oats, and corn are rarely eaten without pre-digestive treatment. Processing extends the lifespan of food. During non-growing seasons, both hunter-gatherers and agriculturalists rely on stored food. Much of the world would have remained uninhabitable if humans hadn't perfected storage. People who live at high latitudes or elevations or have refrigeration can preserve food for some period by keeping it cold. Otherwise, it must be dried, salted, or smoked, and then secreted in a protected place so vermin don't get to it first. The evolutionary legacy of softening and breaking down food left its testimony on the human body. Over time, the thick, massive teeth of early Homo ancestors were replaced by much smaller, more refined, thinner enamel teeth. Robust skulls with bony protuberance that anchored chewing muscles gave way to more delicate faces and jaws. As our heads functioned less to buttress a built-to-jaw mastication system, they made room for brain expansion. Eating partially pre-digested food also relaxed the need for a large and metabolically costly gastrointestinal tract, evidenced by our relatively small gut. Eating pre-digested food also saves time that would otherwise be spent chewing. Chimpanzees spend almost half of the daylight hours chewing highly fibrous fruits, a substantial part of their diet. Humans today spend far less time chewing, about 5% of their day, or about 35 minutes. This dramatically furthers decreases with industrially processed and highly refined food that has little to no fiber. Humans, for most of our existence, lived as hunters and gatherers. Today, only a small number of societies still rely on foraging. But studying how these groups adapt to different environments helps evolutionary anthropologists understand how our species became so widespread and successful. Women's cooperative work. The excitement is palpable when hunters bring meat into a Pume camp. Like many foragers who live near the equator, the Pume live in small groups of about 70 people, relocating their camps and building shelters as the seasons demand. Times are good during the dry season when food is plentiful, but during the rainy season, life changes dramatically. Once a week or so, hunters return to camp with a caiman, anteater, deer, or large bird. I wonder what anteater tastes like. They seem like they have a lot of fur to them. Is there much meat there? I mean, if you're hunter-gathering, you eat what meat you can find. I'm just curious what they taste like and how much meat is actually on them. While meat is valued for its protein and fat, plants get the pume through the wet season. During this lean time of the year, women and children forage almost exclusively for roots and bring in about 85% of the calories. In most temperate and equatorial environments like this one, roots, tubers, seeds, beans, nuts, fruits, and berries are all staples for survival. The Pume women and children supply these bottom line calories and process them into edible foods. So we have a young Pume woman sits beside a hearth processing plant fiber used to construct various tools, such as the fire fan on her lap, the hammock, the ropes behind her, and the basket beside her. So, okay. Simply put, there is not enough time in the day for any one person to forage for plants and animals, process them, haul water, chop firewood, find the raw materials, and manufacture the tools needed to procure and process food, while also finding time to construct clothing and shelter and take care of children. Fulfilling these basic needs requires cooperation. Humans do best by dividing up by age, ability, gender, and sharing the fruits of our labor. I mean, yes, I don't have to go out and hunt or forage, really. I go to the supermarket, done by a bunch of people that I've never met, shipped to my local store so I can buy it, and I get to focus on things like doing this or my job instead of focusing all my time and trying to get food and clothing. It's quite nice. The Pume are not unique in this regard. For hunter-gatherers, food processing is often a multi-step daily task that women and children do together. Hunter-gatherer children may stay in camp while their mothers are out collecting food, but they're not idle. Kids from a young age hang out together, cracking nuts, roasting roots, cooking, carrying water to camp, gathering firewood, and making their own simple tools. Lots to do, lots to do. Although details vary widely across cultures, women in contemporary hunter-gatherer societies typically spend about 20% of daylight hours, or about 3 hours a day, processing food. In most societies, women and children collect the water and firewood that are essential to food processing. Women also manufacture and fix many of the tools used in their work. Pume women, for example, spend nearly 2 hours a day stripping palm fiber, weaving burden baskets, and setting mats, sharpening digging sticks, and hollowing out gourds. A Kong woman in the Kalahari Desert spend around an hour a day making and fixing tools. I wonder why they only spend an hour, whereas this group spends 3. What's the difference? What are they doing? Anyways, the food processing revolution. Food processing has been part of human adaptation since deep in the past. Aside from the earliest known tools in our direct hominin lineage, some 3.3 million years ago, other forms of technology specifically point to the vital role of women and children. For example, the earliest identifiable hearts date to about 300,000 to 400,000 years ago. The stone tools and debris associated with them are often assumed to belong to men, but observational data collected by anthropologists living with contemporary hunter-gatherers tells a different story. Women spend significantly more time than men by the hearth, processing food, cooking, and making the tools to do so. Among the Pume, for instance, 84% of the activities around hearths are of women processing food. This raises the question, is much of the archaeological debris deposited by hearths the byproduct of women's work? See, it's dangerous assuming who's doing what. You don't know. You really don't know. We shouldn't assume when we have no direct evidence of the work. Just that, hey, people did this. While hunting narrative dominated anthropological accounts of foraging societies, women have typically played a major role in food processing. Take, for instance, hunter-gatherers living across the North American Great Plains for millennia. In late summer, these groups would gather to prepare for a bison hunt. Yet a closer look at early ethnographic descriptions of these camps reveals that they were timed around berry picking and women's collective work needs. No? Berries are a key ingredient in pemmican, a calorically dense, highly processed power bar of sorts made of rendered fat, shredded meat, and dried berries that was critical to get people of the Great Plains through the winter months. The unsuspecting berries, which are a natural preservative, are a critical component. Berry picking required a large female labor corps to harvest and process the berries quickly after they ripened and before birds or other scavengers got to them. Pemmican is still an important overwintering food, though market alternatives exist to procure berries. Anybody who's grown berries know that these little scavengers, man, you gotta fight them. The birds, man, they're quick. They get them. If you grow berries, you have to be very on it to get the ones that are when they're ready. Or before they, mold. You don't want that either. Berries mold so quickly. Hunting usually takes center stage as a catalyst for the emergence of food sharing, the division of labor, pair bonding, and joint parenting. Cooperative traits that come to define humanity and sent us down a very different evolutionary path than our non-human primate relatives. But the primacy of hunting is misplaced. Even meat, unless it's pounded, cooked, or otherwise broken down, is difficult to tear off the bone, chew, and digest. The next time you're settling down for a meal, take a moment to appreciate the food processing revolution, the pounding, cooking, mashing done by women. That opened humans to a diversity of food that allowed us to thrive in all of the world's environments. Nice. Yes, cooking! Cooking is good. Leads to deliciousness, and then you all sit around and enjoy. I have very fond memories of spending time learning to cook with my mother and my grandmother, and it was a great time. Moving on. Next. Ooh, another John Hawkes. From August 7th. Hominins voyaged to Sulawesi before 1 million years ago. New report of stone artifacts from Callao place human relatives in Wallaki more broadly and earlier than anyone knew. So we got a picture of stone artifacts from Callao, Sulawesi, images by Emma Watt and Mary Ann. In southern Sulawesi, near the Walane River, is a small museum dedicated to display of local fossils of animals and stone tools. The few display cases of the Callao Paleolithic Museum only hint at the rich ancient heritage nearby. Just go and see if I can get a map. Your reference. Ooh. I am going to be back. I am back. Sorry about that, guys. I need like not my countdown thing. I need a little like standby soon thing. Anyway, where was I? Oh, that's what I was doing. I was trying to get a map. Google Maps. There. This. Marking the spot. And here we are. All in the island. Just north of Australia. South of China. I mean, just north. I mean, that's the big step. Anyways. That's where we are in the world. If you don't know where this is, because I'm sure a lot of people don't know. Next. Ah! Where am I? Okay. Okay. Put that back up so I don't lose it. This stretch of the Wallonie has been an area for fossil exploration. Yes. Okay, for fossil exploration from at least the 1940s, when the Dutch archaeologist Robert van Heekeren came looking for evidence of ancient humans. Van Heekeren and subsequent Dutch-led expeditions found stone tools, some of which seemed to be Pleistocene in age, possibly old indeed. Oh no, okay, it's just him asking. It's not saying you can't read anymore. It's just saying please subscribe. Possibly old indeed, yet the true antiquity of such artifacts was hard to establish with the tools of the era. That changed in 2016 with a description of ancient artifacts from Talepu in the Wallonie Valley. There, Gerrit van den Berg, is this supposed to be Vandenberg or Vandenberg? We're separate, I don't know. Anyways, moving on, sorry about that. Collaborators dug a deep trench and established that stone artifacts from the site date to at least 1,1800 I read that completely wrong. 118,000 years ago. Sulawesi, like Flores and Luzon, had been occupied by hominins long before modern people arrived in the region. Now, our knowledge of the length of the Pleistocene occupation of Sulawesi has grown by an order of magnitude. In 2019, archaeologists identified a stone artifact from a field just a half a kilometer from the Callao Museum. They worked for several years, finding several additional artifacts in excavated contexts, together with teeth from fossil animals that could be dated. This week, the team reports its results in an article led by Budianto Hakim, establishing hominin presence on the island in association with a date of 1.26, give or take 0.22 million years ago. Okay, a quote from Hakim after confirming these stone fragments were indeed artifacts. The field team celebrated the usual way of the Bugis Makassar people with lots of balo, or palm wine. Ah, yes, archaeologists, always celebrating with alcohol. The discovery is as simple as they come. The stone artifacts are simple flake tools, some retouched. There are no hominin bones or teeth, and the team does not report cut marks or other evidence of hominin activity on any animal bones. Still, the presence of stone tools here says much about how early human relatives in the region could adapt and disperse across natural barriers. Sulawesi is just one of many large islands that comprise modern Indonesia, part of the Great Island Arc that stretches from Malaysia to New Guinea. Shaped roughly like a Gothic letter K, the valley of the Wallonie River is in the island's southwest leg. In the mid-19th century, the naturalist Alfred Russel Wallace noted that Sulawesi, Flores, and other nearby islands have a unique combination of animal species. Large islands in the western reaches of the archipelago, like Java, Borneo, and Sumatra, share species with mainland Southeast Asia, including rhinoceros, tigers, and gibbons. Papua New Guinea and Australia have a very different set of animals, including a wide variety of marsupial mammals. We like marsupials, they're interesting. Sulawesi and Flores have few terrestrial mammals, with a handful related to Asian mammals, and in the easternmost islands, like Ceram, some marsupial relatives of species from Papua or Australia. Wallace considered it probable that a deep sea channel had formed. Hair in my eye. A deep-sea channel had formed a barrier to the dispersal of animals from the Asian mainland into Lombok, Flores, and Sulawesi. Wallace recognized the Philippine Islands, with many endemic species, to be a separate situation, but his contemporary Alfred Henry Huxley thought these islands also were part of the same biogeographic history. Today, biologists call these and nearby islands Wallacea. Wallacea. Pleistocene sea level fluctuated to levels as much as 130 meters below today, connecting many land masses that are islands today. Wallace's deep water channel really did exist, separated Sulawesi, Lombok, and Flores from Asia. This kept most terrestrial mammals from invading these islands, except for species that could swim or raft occasionally across the barrier. A history of discovery. Archaeologists who investigated these islands started in the early 1900s, found artifacts made from river cobbles, choppers, and flake tools, in contact with fossils of Pleistocene animals that are now extinct. Van Heekeren's explorations on Sulawesi after the Second World War contributed to this research. He was building on an earlier record of excavation of cave sites in southern Sulawesi, starting in the first decade of the 1900s, and a later Dutch expedition in the 1970s. In the Philippines, parallel investigations were carried out by H. Otley Beyer, starting in the 1920s and 1930s. Beyer worked widely across the region, including the Cagayan Valley of northern Luzon, known today for the 700,000-year-old Kalinga paleontological site. He suspected that many of these simple choppers, chopping tools, and unretouched flakes that he found represented Middle Pleistocene habitation of the island. The most prescient of these early investigations were carried out by the archaeologist and missionary Theodore Voorhoven. Voorhoven worked in the Soa Basin of Flores during the 1950s, finding stone tools and extinct species, including the fossil elephant relative named Stegodon. He also initiated work in Loang Bua during the early 1960s. Some of these finds were happening at the same time as major hominid fossils in nearby Java. By the 1960s, anthropologists were grouping most early Javan fossils into the species Homo erectus. Archaeologists working in the region did not assume that early humans like H. erectus were incapable of reaching Sulawesi, Flores, or Philippines. Indeed, they assumed that occasional crossing of Wallace's line must explain their findings, but none of the work unearthed clear evidence of H. erectus or any other early hominin relatives. By the 1970s and 1980s, many archaeologists based in North America and Europe took on a hardened attitude of skepticism about the dispersal ability of H. erectus and other Pleistocene hominins. This attitude epitomizes the culture last. Restart. This attitude epitomizes the culture last assumption that I wrote about last month. Following this assumption, ancestral humans should have had the same limitations as most large Asian mammals. They couldn't swim across the straits that separated Wallacea from Java or Borneo. They had no boats until modern humans appeared in the region, sometimes after 70,000 years ago. Following this way of thinking, the hominins like Homo erectus, Flores, and Sulawesi may as well have been the moon. The discoveries from the Liangbua Flores, including the fossil bones attributed to... that scared me. Does that every night. My sprinkler needs to be adjusted and it hits the window that's right here, and it scares me every night. Anyways, the bones attributed to H. floresiensis shifted perceptions by showing that other hominins were likely present in Wallacea. At first, the true age of the Liangbua evidence was not recognized, where the most complete finds argued to be less than 35,000 years. That would change. More important, teams of archaeologists and paleontologists began to reassess. I'm so sorry if you guys can hear that. More important, teams of archaeologists and paleontologists began to reassess evidence from a wide range of sites across the region, from Luzon and Sulawesi as well as Flores. From Liambua and the Soa Basin to Calao Cave and Kalinga to the Calio and Talaku, recent archaeologists are revisiting regions where discoveries were made and published more than 60 years ago. They keep finding that there is vastly more to find, particularly with today's improved geochronological methods. These current discoveries are emerging because of the interest and investment 3 decades ago by researchers who wanted to know if Verhaven's discoveries could be replicated or exceeded. These investments keep paying off for the entire world, the entire field, not the world. Well, I guess the world's benefiting from it. Knowledge of our past. The importance of Calio. I've seen a few people asking if Sulawesi is going to produce another species of small hominins, similar to what is observed of H. floresiensis and suspected of H. luzonensis. I doubt it. Cox doubts it. Sulawesi is larger in area than Flores or Luzon, and it sustained a more diverse community of large mammals, including 2 extant species of anoa and a possible fossil ancestor, a dwarf stegodont. The extant babirusa, a warty pig, and an extinct pig, Celebopterus. The continued survival of most of these species after the arrival of modern humans may suggest that they co-adapted even within a faunal community that included hominid predators more formidable than Homo floresiensis. The great diversity of large mammals may also owe something to geography. Sulawesi also shares a much larger coastline with the Strait of Makassar. Looking across at Borneo, the crossing is longer than the Bali Lombok crossing toward Flores to the south. But with prevailing currents, someone who set out toward Sulawesi may be less likely to miss the island entirely. Island hopping from Mindanao to the north may not be out of the question. This geography makes it plausible that hominins entered Sulawesi from different geographic sources repeatedly. The geography is not new, of course, and it is why I still expect Denisovans likely occupied the island. I interpret later Middle Pleistocene or Late Pleistocene record, including Talepo, with that phone is going off. With that idea in mind. But Denisovans did not exist a million years ago. The Denisovans, Neanderthals, and African Idaltu branched from each other around 700,000 years ago, making the earliest possible Denisovan presence in the region much later. In fact, the date that Hakim and co-workers present for the Calio material has an upper error bar stretching to 1.48 million years. If these tools are actually close to this upper extreme, they are within error of the oldest known hominid fossils from Java. The geochronology of Sengirin and Mojokerto has always been disputable, but recent results suggest that the first appearance date of early H. erectus on Java is less than 1.5 million years ago. Based on these numbers, Calio could be even earlier than H. erectus on Java. Was first colonized from the east? Oops, little typo. Not calling on his little stream here. Although it would be far from the first time, a 500,000 year error range was the hanging point for a theory. It's a thin hook to support one. I wouldn't go there. Instead, here's what I would suggest as a shift in perspective. Hominins in island Southeast Asia had behavioral familiarity with shoreline resources and crossing deep rivers from the start. A set of strategies that Carlos Neto de Carvalho and collaborators have recently called coast steering. Their early hominins in this region found ways again and again to exploit rich coastal ecosystems, including mangrove forests, tidal pools, riversides, and nearshore islands. It is not possible to develop this array of foraging strategies without becoming familiar with swimming, interacting with floating materials like logs and vegetation mats, and constructing simple watercraft. Sea level fluctuations in islands in Southeast Asia sometimes happened on decadal scales. Oh man, I think this is my last article. Creating new near-shore islands with once connecting land masses. The dispersal potential of hominid groups with the ability to employ simple watercraft was greater at every water crossing compared to land-limited groups. Geography selected for water familiarity throughout this region, if not long before. I expect that Sulawesi saw dozens or hundreds of hominid arrivals once they arrived in the region. Every newcomer would have had a crack at the island at some point, from early Age erectus to Denisovans, to the first modern people, to Mesolithic and Neolithic immigrants, to historic times. The question in my mind is whether during some long span of the Pleistocene, these newcomers had enough numbers to constrain the genetic and cultural evolution of the Sulawesi hominids, or whether instead Sulawesi, as is possible on Flores and Luzon, may have developed a population with sustained numbers and adaptations that differed from new arrivals. It will take many more discoveries to find out. Time will tell. Ooh. Nice and depth. Anyway, I think I'm going to end the stream there. I am not reading very smoothly anymore. Not that I ever read very smoothly, let's be honest. I do hiccups all the time, and half the time I can't read the words. Reading out loud. Yay. Anyways. Thank you guys, whoever watches this. And I will see you guys next time. Hopefully it won't be too long until next time, but we will see. You guys have a wonderful evening, and I am so sorry for the sprinkler if you can hear it. I hope you can. I'll listen to that, see if we can. If so, I will avoid that. I just need to go reset it. Shift it down so it doesn't hit the window. You guys have a wonderful evening, and take care.